So let's again focus on the peptide itself, just a simple peptide. And thinking about its structure and naming these peptides based on what we know from amino acids. Now, first, the ends of a peptide are not the same. As you add amino acids, you form these peptide bonds. And again, those are highlighted here. This is a peptide bond that links together the first amino acid 1, and the second amino acid 2, and so on and so forth as we link on more amino acids 3, 4, and 5. Now notice we have two ends of our peptide. One end is called the amino terminal end. That is the end where the amino group that has not been linked sticks out. This is an NH3 plus group. The other end is the carboxyl terminal end, where our last COO minus, again unlinked, the end of the chain, is sticking out. When we read amino acids, we always read them from the amino terminal end to the carboxy terminal end. So anytime a name is given or the peptide code is given, it will always be from the amino to the carboxyl end. And this is also always how you should draw them out. If you're drawing a peptide, they should be drawn functionally, starting with the amino terminal end and ending with the carboxyl terminal end. Now notice here, this is a peptide of multiple amino acids that have been joined together. And I've kind of highlighted where they are. And you can see in this image in pink, the special R group that makes each amino acid is highlighted. So we can see that dissecting this up, you can see our original first amino acid here, and it goes all the way essentially to here, where it is then joined by the next amino acid by a peptide bond, and so on and so forth. This one's all the way up here. But generally, the structure of the amino acid is the same. You can see where the central carbon is. You can see the hydrogen that the amino acid has, that all amino acids have. You can see where the amino group is. You can see where the carboxyl group is. And when it's forming a peptide bond, you can see what is left of the carboxyl group. And thus, the last position is still held by the R position. So looking at a chain like this, you can tell what each amino acid is based on that special R group. So our first one here, the CH2OH, we should know after memorizing our structures of amino acid that this is serine, S. The next one, just a hydrogen, is our simple and first amino acid, glycine, G. The next one, our ring, tire-looking amino acid, tyrosine, Y. The next, just a methyl group, alanine. And the very last one, leucine. This is how you can take a peptide chain and dissect it into each of the amino acids and figure out the code of a whole peptide chain. Now, if you were to name this particular peptide chain, there are numerous ways to do it. One way is by using the full amino acid names. You would take the name of each starting amino acid with a YL ending on it. So serine becomes seryl, glycine becomes glycol, tyrosine, tyrosol, and then alanine, alanol. The very last amino acid in the sequence, the amino acid that is at the carboxyl end, is written normally without the YL ending. So as you can see, the name of the sequence previously was seryl, glycol, tyrosol, alanol, leucine. Another way to write this is using the 3-letter code abbreviation. We didn't get as deep into using 3-letter codes, but you can see the representative 3-letter codes for those amino acids. Again, serine, glycine, tyrosine, alanine, and leucine. Now, when you have a very, very long peptide, we tend to shorten this even further to its 1-letter code. And that's why we use the 1-letter codes and why I had you memorize the 1-letter codes. Generally, when you have the whole structure, amino acid structure of a protein, which can be very long, we write it in these terms of 1-letter codes. So here, the 1-letter code for this short peptide is SGYAL, just as I showed here. And again, you always write this from amino end to the carboxyl end. So let's do another practice. Let's name this particular amino acid. Again, we want to break this down into the amino acid subunits, find the R group, and then write out the letter code for each of these amino acids. You can see in this practice, it's already written with the amino group on the left and the carboxyl group on the right. Perfect. That's the way that our structure should be written. If you do get a problem that doesn't have it written this way, you should first rewrite your structure so that it is amino group to carboxyl group. Next, we're going to kind of highlight out where each amino acid is. You can draw a box or you can circle them, or you can put a breakpoint to show where each amino acid block is. Remember, in between each peptide bond, these are the linking bonds, is where we have our amino acid units. So there are 4 amino acids here. 1, 2, 3, 4, and each of them have their own unique R group. Based on the R group, we're going to decide which amino acid this is. So this R group here on the first one is for a glutamine. You can see here's the amide group that makes up glutamine, and that letter is Q. The second group here is a lysine. This is another charged amino acid with an NH3 plus at the end, and lysine's 1-letter code is K. The next group is histidine. It's this ring group made of a cyclical heterocycle of carbons and nitrogens. Again, it has a positive charge. This is H. And our last is a polar amino acid, CH2OH. This is serine. So our full name, if you wrote it in 1-letter codes, would be Q, K, H, S. Now let's try to do the opposite. What if you're given a 1-letter code and asked to draw a structure? Well, just like before, I want you to remember to always start from the amino group end, and we're going to end with the carboxyl group. So we should start with an NH3 plus here. Here is our first central carbon amino acid block. Remember, every amino acid has a hydrogen on it. Here's the amino group. And we're going to start with our carboxyl group. This is going to form a peptide bond because we're bonding these together. And our R group will be here. I'm going to continue the peptide bond. We know that every time we link two amino acids, we have to have this peptide bond of a C double bond O and an NH. And that will start onto our next amino acid. Again, here's the central carbon for that amino acid. I'm going to draw its hydrogen and a spot for its R group. And again, we'll draw the linking peptide bond. This is that peptide bond. So we have one central carbon and then two central carbons. Notice we have we have 4 amino acids we have to draw in. This is our third central carbon. Room for its R group. Another peptide bond because we have one more carbon to add. And here is our last central carbon. Now we're on our last amino acid. So add the hydrogen as normal, add a space for the R group. Now this one, because it is the last amino acid, we're not linking another peptide bond on it. It will end with our COO minus group. Okay, so now let's add in the R groups based on our given amino acid code. We've drawn out essentially a peptide backbone. This is how you would draw out your peptide backbone. And here's each of the peptide bonds that links all of our amino acids. So our first amino acid, R group we need to draw is for G, which is glycine. Glycine's R group is just a hydrogen. The next one, C, is for cysteine, which is the CH2SH. The next is D, which is for aspartic acid or aspartate. That is CH2, C double bond O and O minus. And our last one, K, is for lysine. So again, you just have to memorize these structures to add the R groups onto each of these. And there you go. That is how you would draw out this particular peptide, GCDK.